Okay, everyone, welcome back to part 2 of the video lecture for Chapter 7 on membrane structure and function. At the end of part 1 of this chapter, or the video for this chapter, we had talked about membrane selectivity and how it is that phospholipid bilayers, cell membranes, act sort of like filters, preventing certain things from moving through very easily while allowing other things to move through quite easily. But in all those cases, we were talking about solutes, for example, ions, sugar molecules, fat molecules, whatever, just moving down their concentration gradient, moving from an area where they're in high concentration, maybe outside the cell, across a membrane to an area inside the cell where they're in low concentration. So that's called diffusion or passive transport. Things do this. They move from high concentration to low. Okay, they don't go back from low concentration to high unless, of course, you apply energy. But they do move from high concentration to low with no additional input of energy. Usually it's the heat already present in the system that drives that movement from high concentration to low. And again, that's called passive transport because it requires no energy. Passive implying no work required, no energy required. All right, now we're going to talk about, kind of take that one step further and talk about a special case of passive transport. That special case is osmosis, all right? Now, osmosis is what you're seeing an example of right here, and I'll explain this figure in just a moment, but for now, let's define osmosis, all right? So, osmosis is simply the diffusion of water across a selectively permeable membrane. So osmosis is diffusion, it's passive transport, but specifically of water. Okay, so here we see another example where, because water is a very special substance, we kind of set aside a special place for it or a special category for it. Osmosis is diffusion of water down its concentration gradient. And this can actually have or be a very powerful force in terms of the way that it affects cells. So if we look at this figure from your textbook, okay, what we can see is a good example of how osmosis can affect cells. So let's imagine that we have this U-shaped piece of glassware and the two halves are separated by a selectively permeable membrane. If you zoom in, you'll see it's selectively permeable in the sense that it has openings in it, but those openings are a specific size. So it's selective in the sense that anything that's too large to fit through the openings can't go through the membrane, and vice versa. Okay, so we have two sugar solutions, one on the left and one on the right. The little green circles are sugar molecules, okay, representatives thereof. So here we have a more dilute sugar solution, right? So we have a lower concentration of sugar on the left. Compare that to the right, where it's obvious we have a higher concentration of sugar. If you could taste these two solutions, the one on the left would taste less sweet, and the one on the right more sweet. Okay, but notice that the fluid levels, solution levels, are the same height. There's the exact same volume of solution here as there is here. Same volume on both sides. Okay, so we can clearly see from this example that there's a higher concentration of sugar on the right and lower concentration on the left. Now, we need to use that information to infer something about the water concentration, because again, we're trying to understand osmosis, and osmosis is water moving down its concentration gradient, all right? So to understand why it moves and what direction it does, we have to understand the gradient, the water concentration gradient. So, on the left, we have fewer sugar molecules than on the right. But because the solutions are at the same volume, that means therefore on the left, by default, we must have a higher concentration of water. More water on the left and a lower concentration or less water on the right. These extra sugar molecules here on the right-hand side in the sweeter solution, more concentrated sugar solution, take up space that could be occupied by water. So because they're the same volume, there's less water here, and respectively more water there on the left-hand side. That means we have a higher concentration of water on the left, so water will tend to move down its gradient from left to right, assuming it can get across that membrane, okay? And it can. The water molecules can fit across the openings or through the openings in that membrane, so water selectively moves to the right. Most of the water molecules, as you see, are moving to the right. Some may go to the left, but they're outnumbered by those that are moving to the right. That's water moving down its concentration gradient. That's osmosis. Remember also I said in the previous video for this chapter that nature tends to eliminate gradients. Nature doesn't like gradients, okay? Gradients represent imbalance in nature, basically. So we also have a sugar concentration gradient. Higher concentration of sugar on the right, lower on the left. So sugar also wants to move down its gradient, but it wants to go from right, where it's in higher concentration, to left, where it's in lower. But take a look. The openings are too small. The large sugar molecules can't fit through. This means sugar molecules, or the sugar gradient, cannot be eliminated by movement of sugar molecules alone. So what has to happen here is that the water moves from high concentration to low until the concentration of water and sugar is the same on both sides. So if on the right-hand side we have more sugar molecules than on the left, and they can't diffuse to the left to equal out that difference, water has to move to the right to dilute this solution down until it has the same concentration of sugar molecules as you have over here. That means water is moving, there's more water moving from left to right, and it continues to move until the concentration of sugar and water is the same on the right as on the left. Well, because of this diluting down effect, because sugar molecules can't move, we have to increase the volume of solution on the right-hand side to dilute it down until it matches that on the left. And you see, now the solution volumes are different, right? The elevations are different. If this is a cell, that cell may have taken on too much water and could actually burst or rupture, lyse as we say, and tear open, and that could kill that cell. So in this way, osmosis can be a very powerful force, all right? Okay. So we have a number of terms that we use to describe these situations regarding concentration gradients of water and solutes, all right? First one is a more general term, tonicity. Tonicity is the ability of a surrounding solution to cause a cell to gain or lose water, all right? So tonicity just generally describes the concentration gradient situation of solutes inside and outside of a cell. Now, we basically have three different situations regarding concentrations of solutes inside and outside the cell. First is isotonic. So prefix iso means same. Tonic refers to solute concentration. So in an isotonic solution, or let's say we have a cell sitting in an isotonic solution, that means that cell has the same concentration of a particular solute inside as you find outside. And that solute is going to be in an aqueous solution, right? A water-based solution where water is the solvent. So if we have the same concentration, let's say, of salt inside the cell, in the salt solution inside the cell, and the same concentration of salt outside, that also by definition means you have the same concentration of water inside versus out. So we have the term isotonic tells us we have no concentration gradient for the solute. That by definition means we have no water concentration gradient either. So if there's no water concentration gradient, water doesn't move into or out of the cell at a net rate, right? The cell does not gain or lose water. If one water molecule goes into the cell, it's balanced out by one that leaves, and so the concentration of water doesn't change. The cell does not gain or lose water. Now, what if we have a hypertonic solution? So we have a cell sitting in a hypertonic. Hyper means a lot or great, right? Okay, think hyperactive child. So if we have a cell sitting in a hypertonic solution, that means the solute concentration is greater outside the cell than inside, right? The solution outside the cell is hypertonic. To the cell inside, there's a higher concentration of solute outside the cell than inside. That means the situation for water concentration is the reverse. In this case, you'd have a lower water concentration outside the cell than in, or greater inside than outside, and so water would tend to move down its gradient from the inside of the cell to the out. A cell placed in a hypertonic environment will tend to lose water because the salty solution that it's sitting in will tend to draw water out because water is trying to move out to eliminate that water concentration gradient and also the solute gradient. So another way to think of this situation is water moving toward higher solute concentration. Here you have higher solute concentration outside the cell. Water moves from inside to out. So water's moving down its own gradient from high water concentration inside to low water outside, but it's moving toward higher solute concentration outside. Finally, we have hypotonic. So if you have a cell sitting in a hypotonic, that means that there's a lower concentration of solutes outside the cell. Hypo means little, less, or low. So here the solute concentration is outside the cell is less than inside, which means the water concentration outside the cell is greater than inside. So the water concentration situation is always the reverse of the solute concentration. But these terms refer to the solute concentration. So once you understand what these terms mean for solute concentration, you flip the situation around to the opposite, reverse. That tells you what's going on with the water concentration situation and helps you understand which way water flows, if it does. All right? In a hypotonic situation, a cell placed in a hypotonic solution, in other words, will gain water, and that cell might actually rupture or tear open or lyse. So let's take a look. So here we have cells in three different situations, okay? Animal cells as a blood cell on top, plant cells on the bottom. So let's look at hypotonic first. So hypotonic. All right, again, hypotonic means that the solution the cell is sitting in has a lower concentration of solutes than inside the cell. That means greater water concentration outside the cell. So water will tend to move down its gradient from outside the cell to in. And in the case of an animal cell, which just has a cell membrane and a relatively flimsy ECM on the outside, if it takes on too much water, it will lyse, it will tear open, and that will be the death of that cell. Now, plant cells, having a rigid cell wall, a suit of armor around them basically, don't have that problem. They'll take on excess water from the outside, but only up to a point. Once the water pressure inside is greater than the water pressure outside or equals it, water intake stops, and we say that cell is turgid, has high water pressure inside of its cytoplasm and also in its central vacuole. That's a turgid plant cell. Plant cells are healthiest under those conditions. That's what happens to plants when you just water them, or when they just take up soil or water from the soil after it's rained, for instance. If you ever have lettuce or celery that you pull out of the fridge and it's kind of shriveled and wilted, or the celery bends rather than snapping, soak it in water for a little bit and it will take on excess water, and those cells will become turgid and it'll seem crisp again. Okay, both the lettuce and the celery will do that. It's because the cells take on water. You put them in a hypotonic environment and they go turgid, which is the healthiest situation for a plant cell. Now, what about isotonic? Iso means same again, so now we have the same concentration of solutes outside versus in, which means same concentration of water outside versus in. So the amount of water that's going into a cell is balanced by the amount leaving, and so the cell does not gain or lose water. Animal cells are healthiest or normal in that situation. That, however, is the point where a plant cell becomes what we call flaccid. That means the plant cell essentially lost enough water that the pressure has dropped inside of it. It's no longer turgid, it's flaccid. This is the point where a plant may begin to wilt. Plant cells really aren't very healthy like that. If that goes on too long, they can actually die. All right. Now, what if we put either of these cells in a hypertonic environment? So now we have hyper, or a lot of solute outside the cell, less in. That means you have more water inside the cell than out. An animal cell will lose water and it will shrivel up as a result. If that goes on long enough, that can kill that cell. In the case of a plant cell, which again has that rigid cell wall, it won't shrivel up like an animal cell will. Instead, what happens is something called plasmolysis. All right, let me write that down. Plasmolysis, in other words, or plasmolysis as we say. This cell has plasmolyzed. In other words, the cell membrane, which is normally sort of pasted up against the inside of the cell wall, contracts as water leaves, and it really doesn't look like this. It doesn't stick like you see there. It sort of condenses down or shrivels down to a sort of sphere or an egg-shaped structure inside the cell. And all the guts of the cell are now inside of that cell membrane, which has lost water and shriveled down around it. You can see this pretty clearly in plant cells. Okay, you take a drop of salt water and put it on a piece of plant tissue on a microscope and watch what happens under the scope. You can see this plasmolysis, and I actually have a video of that right here. This is in slow motion, there's no sound to it, but you can see what happens. All right, so right here, for instance, you can see that cell membrane, okay, pulling away. You can see it here. You can see it right there, for example. As it continues on here in slow motion, it gets even more pronounced. And eventually you start to see what looks like a bubble of cell membrane inside each of those brick-like cell walls, okay? Pretty clear, especially right here, for instance, right? You can see the cell wall, or cell membrane rather, has pulled away from the cell wall. There's a cell wall, and all the contents of the cell are now sort of condensed down inside that membrane. Those plant cells have been put in a hypertonic environment, and they've lost water. That's plasmolysis in a plant cell. And if that goes on long enough, that can kill the cell, as we said. So this concept of tonicity, isotonic, hypotonic, hypertonic, can have really dramatic impacts on cell health. And if things go too far out of balance with regard to water or solutes, you can have cells dying. This is also, for instance, why you see this at the grocery store, why they spray the produce with water every so often. It's because they want to keep water on those leaves, on the surface of that produce, to keep those cells turgid and to keep the vegetables and things looking nice and crisp and that sort of thing, fresh. Okay. All right. So, how is it that organisms maintain relatively constant internal environment with regard to water, solutes, and things like that? Well, the processes that organisms use to do that are complex and varied. There are quite a few of them. But overall, they're referred to as osmoregulation. So the different mechanisms that organisms use to control solute concentrations and water balance are referred to as osmoregulation. Excuse me. And so you can think of, for example, freshwater organisms, especially freshwater single-celled organisms. They're living in a hypotonic environment. They have an overriding problem to deal with every second of their lives, and that is water is constantly flooding in from outside to in. If they don't have some way of getting rid of that excess water, they're going to lyse, they're going to tear open, and that would be the end of that cell. So the various mechanisms that cells use for that are referred to as osmoregulation. So a good example of that is the paramecium. This is a single-celled organism. Maybe you saw this, in high school biology lab when you looked at pond water or whatever. But this is the paramecium. You can see that fringe of cilia around that. And I think I showed a video of one of these in the last chapter. What it has is two contractile vacuoles. There's one here and one right there. You can see this one a lot better. This contractile vacuole is made of cell membrane, phospholipid bilayer, and it has these arms that radiate out from it in sort of like a star pattern. Those increase surface area of the membrane of the vacuole, and it keeps it in contact with the cytoplasm. So as water continually rushes into that cell because it's hypotonic outside, these arms collect that water and funnel it down to the contractile vacuole, the center of it, which eventually fills up, and then the cell ejects it. It's sort of like it's bailing water. It squirts that water from the vacuole to the outside. This is how it maintains balance. It sort of offsets the water that's coming in by constantly pumping water back out. You can kind of think of it as sort of like a bilge pump in a boat that keeps any water coming in from leaks in check. All right, works pretty well. And then there are lots of other mechanisms like this for various different kinds of organisms. Okay. All right, so that's osmosis. The next thing we're going to talk about is what's called facilitated diffusion, and that's what this figure here is about. So let me write that. Facilitated diffusion. All right. Now, one point to clarify. We're still talking about diffusion here. Okay, all the processes we've been talking about so far are passive in that they don't use energy. That was true of simple diffusion. That was true of osmosis that we just finished talking about. And it's true of facilitated diffusion as well. But here in the case of facilitated diffusion, we're talking about solutes that, let's say, are polar or hydrophilic, and therefore they can't get across that internal fat layer, hydrophobic layer of the cell membrane. There's a channel protein of some sort, or another kind of protein, a carrier protein we'll see in a minute, that allows a route of safe passage for solutes from high concentration to low across that membrane. So it's diffusion because we're still talking about solutes moving from high concentration to low, but it's facilitated diffusion because there's a protein helping this process along, helping a solute move through the membrane when it can't get directly through the membrane because it's hydrophilic or polar. All right, this is the simplest case. You have a channel protein just acting as a tunnel to allow solutes to move through, in the same way a tunnel allows you to drive through a mountain on the interstate. Here we have a little more special case in the bottom of facilitated diffusion. Here we have a situation where you've got a solute that has a shape that allows it to fit specifically into a protein, what's called a carrier protein. Think of a carrier protein as kind of like a revolving door, right? So because of the shape recognition between these two molecules, the solute and the protein may only move that particular solute. That solute can come in and bind because it has the right shape. When it comes in and binds to that site, the carrier protein changes shape and sort of inverts or passes that solute through in that way. This is one way, again, that proteins can be selective and not allow just everything, any old thing to move through. Maybe the carrier protein moves only one thing. It's much more selective than what you see here with the channel protein, where anything that can fit can move through. Okay, anything that's small enough, in other words, to pass through the tunnel, basically. All right. We also have a special category of membrane proteins called aquaporins. Now, aquaporins, think of them as sort of like floodgates, okay? They are channel proteins that specifically allow for movement of water. So when a cell needs to get a lot of water across its membrane quickly, either in or out of the cell, it can open up these aquaporins like floodgates and allow water to flow pretty quickly, okay? We also have what are called ion channels. We won't talk a whole lot about these, but ion channels facilitate diffusion of ions, specifically like sodium ions, potassium ions, that kind of thing. Some of them are called gated channels in that they open or close in response to a nerve impulse or some other sort of stimulus, okay? But again, these are just different ways for the cell to control what moves into and out of the cell, all right? So, again, these carrier proteins that you see here are going to undergo a subtle sort of shape change or conformational change that'll translocate that solute across the membrane. Translocate means to move something across the membrane or across a space or a distance. Okay, so facilitated diffusion, as we said, is still passive because the solute in every case is moving down its concentration gradient, so no additional energy is required to make this happen. But now we're going to shift gears a little bit and talk about active transport. So active transport, or the term active, refers to the fact that energy is involved here to move solutes. And the reason why energy is necessary here is because active transport is involving the movement of solutes against their concentration gradient. So from low concentration back to high. That is always going to require energy because you're working against the natural tendency of things to diffuse from high concentration to low. All right. So, so this is an active process requiring energy. Okay, so what is it that provides the energy for cells to carry out active transport? Primarily, it's a molecule called ATP, okay? We'll talk more about ATP, especially starting in chapter 8, the next chapter, but for now, just think of ATP as a molecule that carries energy. It's like a fuel tank. ATP provides the energy that allows specific proteins to pump solutes from low concentration on one side of the membrane across the membrane toward high concentration. So active transport is a way or a method that cells use to maintain concentration gradients, which is extremely important for cell chemistry and the way that cells function. We're going to see lots of examples coming up in the next few chapters of situations where cells require solute gradients to be present. And they can set those up themselves by pumping things from low concentration to high and maintaining those gradients, but it does require energy in the form of ATP typically. And these proteins that are embedded in cell membranes that are going to perform active transport are going to act like pumps. You can think of them as membrane pumps or active transport pumps. All right, one really good example of this is what's called, especially in animal cells, what's called the sodium-potassium pump. All right, the sodium-potassium pump. This is a really important active transport pump in animal cells that allows the cell to maintain two concentration gradients simultaneously, one for sodium and one for potassium. All right, so let's take a look. This is an illustration showing how the sodium-potassium pump works in sequence, all right? So first of all, the sodium-potassium pump is this purple thing. That's our protein. That's the pump itself. Inside of it, it has 3 sites that accommodate or bind sodium ions, Na+, and 2 sites that bind potassium ions, okay, K+. Now, we're going to start with sodium here. So when the when the protein is open to the inside of the cell, sodium from inside the cell, where sodium is in lower concentration, can come in and bind to these sites in the sodium-potassium pump. So once that protein is then loaded with sodium, an ATP molecule comes in, and the third of the 3 phosphates, adenosine triphosphate, right? The third phosphate breaks off and attaches to the protein. Think of that as an injection of energy from the ATP molecule. So now that protein, which is loaded with sodium ions, has the energy it needs to pump them across the membrane. And so we say that that protein is phosphorylated. Phosphorylated. Okay, that just means that it has a phosphate group from ATP attached to it, and now it's higher energy than it was before, or it has more energy. So once the phosphate group attached to it, you can see that the protein changes shape, closes up on the inside, opens up on the outside, and passes the sodium ions to the outside, where they're in higher concentration. So it's using energy to pump those sodium ions from low concentration inside to high concentration outside, maintaining that concentration gradient of sodium. Now, while it's still open to the outside, while the phosphate group is still attached, 2 potassium ions come in and attach. Then our phosphate group breaks away. And you see how it says P sub I? That just means it's now inorganic, okay? It's PO4, it's a phosphate group. There's no carbon, hydrogen, it's not attached to an organic molecule like this protein, it's inorganic phosphate. It comes away, the energy comes back out of the protein, it goes back to its original shape where it's now open to the inside, closed to the outside, and it sort of ejects the potassium ions to the inside where they are in higher concentration. So they've moved from low concentration outside to high concentration inside against their gradient. So in these two movements where the protein has taken energy from ATP, in these two movements it has pumped sodium against its gradient and potassium against its gradient and has maintained both of those ions in a concentration gradient situation across that membrane. All right, so I have an animation for this. Let me play.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here is how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and push the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
Okay, so hopefully now you have a better understanding of how the sodium-potassium pump works in particular, and more generally how active transport works. So the sodium potassium pump is a classic example of an animal cell active transport pump. Plant cells don't have the sodium potassium pump. They use, for example, in addition to others, they use what's called a proton pump. We're going to talk more about the proton pump here in the last part of this chapter. But real quick, I want to summarize the various modes of transport that we've talked about this far in this chapter. And we're going to let this be the last slide for part 2, and then we'll save the remaining part of this chapter for the third video lecture. Okay, so everything here on the left is a case of passive transport. Again, passive, none of these situations requiring any energy. In every case, whether it's diffusion or facilitated diffusion through a channel protein or facilitated diffusion through a carrier protein, we have some sort of solute moving from high concentration toward low concentration. That's why this is all passive. That does not take energy. The energy to do that work is already present as the heat there in the system. No more energy has to be added, happens on its own. Okay, so simple diffusion, no protein involved, right? Some sort of solute can just diffuse directly through that membrane. Water molecules can do this to a certain extent because they're very small, but to move them quickly, we need an aquaporin, like I said, for facilitated diffusion. Now, other solutes that might be hydrophilic and polar and are too big, let's say, to get through that fat layer of that membrane, will need a channel protein or a carrier protein. They'll need to use facilitated diffusion. Now, in a situation where we're moving some sort of solute from low concentration to high, there we're going to be pumping something against its concentration gradient. That's going to require energy, typically in the form of ATP and a protein pump. So this is active transport, active using energy to pump something from low concentration to high against the natural flow of things, all right? Okay, so that's going to be it for the second video lecture of this chapter. We'll wrap this chapter up with the third video lecture next.